Greetings to all the eminent guests and delegates present here. I'm Supriya, a remote intern working at Dr. Karishma Kaushik's lab in SPPU, Pune, India. I'm here today to present to you on behalf of Dr. Karishma Kaushik's lab on our one-stop structural and functional repository for AMPs and biofilm targets called BAMP. BAMP's initial version dealt with antimicrobial peptides and their properties. Here we are presenting the updated repository called BAMP version 2.0, enhanced functionalities and capabilities in tackling biofilm-induced resistance. We are all aware of the opportunistic infections in hospitalized patients across the world. The growing challenge to tackle antibiotic-resistant pathogens calls for an immediate rescue. The pathogens have various strategies that support their pathogenic resistance, one of which is biofilm formation. What are biofilms? Biofilm is an assemblage of surface-associated microbial cells that is enclosed in an extracellular polymeric substance matrix. When an antibiotic-resistant pathogen infects the host, it adheres and initiates biofilm formation using biofilm-forming proteins. Antimicrobial peptides, or AMPs, are agents released as defense mechanism by all organisms in nature. BAMP version 1.0 was a structural and functional repository of AMPs sourced from various organisms. This data was useful to study the effect of AMPs on protein targets responsible for biofilm formation. Currently, biofilm researchers don't readily have a curated list of all biofilm targets available. A curated list like this can be used to treat and prevent disease proliferation. So here comes BAMP version 2.0, a structural and functional repository of such biofilm targets. To build BAMP version 2.0, we have collected relevant target data from across three databases, PDB, UniProt, and PubMed. To gather the data, we used a custom list of keywords and advanced search features of respective databases using in-house API scripts to collate a metadata of all biofilm targets. Upon removal of redundancy, we obtained a metadata of 2,502 unique potential biofilm targets. Of these, 102 had 3D protein structure available, and select 90 biofilm targets with no structure available were modeled using Rosetta Fold. BAMP version 2.0 has a dedicated tab for biofilm targets where the user can search with keywords and filter the results based on organism name or function. The results get populated in real time as the user types the keyword. The results will be displayed as color-coded tiles based on their PDB-defined function. Each tile contains information such as unique target ID, name of the source organism, downloadable FASTA files, PDB or UniProt Go functions, PDB Qt files, and PubMed links to relevant literature. The biofilm targets on BAMP version 2.0 belong to 470 different bacterial species, of which the highest number of biofilm targets are from Escherichia coli, followed by Salmonella enterica, Staphylococcus aureus, Acinetobacter baumannii, and Klebsiella pneumoniae. Additionally, we have expanded the case study using C. striatum sortes C with LPMTG motif of the Pillin subunit labeled as PEP1 here by subjecting it to a molecular dynamics simulation of 20 nanoseconds. As we conclude, we present the MD trajectories validating the previously predicted preferential binding of select AMPs from BAMP version 1.0 with C. striatum sortes C, a potential biofilm target. The table here indicates the initial and final stages of these 20 nanosecond simulations. For 90 biofilm targets from Pseudomonas aeruginosa and Staphylococcus aureus with no structure available, we modeled their 3D protein structures using Rosetta Fold, and we will be linking these to their respective tiles on BAMP version 2.2. These two pathogens are widely encountered and often observed together as co-pathogens. We intend to model structures for more targets with no structure available and make them usable to researchers for in silico studies. We aim to make BAMP a one-stop repository of AMPs in biofilm targets. BAMP version 2.0 is a great resource for users, especially into biofilm research, drug design, and microbiology. We are grateful to ISCB for making it possible for us to present at ISMB 2022, and we are thankful to Dr. Karishma Kaushik and Dr. Raghottaman Yanamalli for their support, and also their respective funding agencies. Thank you for your time.